So it's going to take us a couple days to talk about meiosis. It's like mitosis. It has prophase, metaphase, anaphase, telophase, but it's not for the same kind of cell, and it doesn't serve the same purpose. So first couple points about meiosis that make it different. One, this is what's happening in germinal cells. Mitosis is what happened for somatic cells. Germinal cells are the ones that are going to produce gametes. So in a human, realistically, the only place we would see this process happening are in the cells that are going to produce sperm cells in males or egg cells in females. No other body cell realistically will do this. The other thing is that it's not a cycle. If a cell went through mitosis, it could go right back into interphase and then go through mitosis again, go back into interphase, go through mitosis again. But meiosis is not a cycle. Once a cell has finished going through all the steps of meiosis, that's it. It's not going to go through meiosis again. And you'll see why when we get to the end of it, but it's not a cycle. Now, there are two reasons that meiosis is important. And the first one is first on purpose. This wasn't a random choice. If a question ever asks you, what is the purpose of meiosis? This is it, reduction division. Meiosis happens to produce cells that have fewer chromosomes, specifically half than the parent cells. So if we're talking humans, our somatic cells have 46 chromosomes, but our gametes only have 23. And we make them through reduction division in meiosis. The second reason that it's important has to do with what can happen afterwards. Recombination is possible. One of the reasons that sexual reproduction is useful is because it produces variety. Organisms are different. They don't all have the exact same traits. And the reason that happens is because the products of meiosis, the gametes, have different combinations of genes. And when they combine, children are genetically distinct. You might notice that you have some traits that are kind of like your mom, some that are kind of like your dad. Maybe some of you have a whole bunch of traits that are like your mom and a couple that are like your dad or vice versa. But you are not identical to either one of your parents. You're similar to both of them, but not identical to either. There's recombination. There's variety. In organisms that don't produce gametes, that reproduce asexually, this doesn't happen. So this is a benefit of meiosis having completed. Now, before we go through the steps that happen during meiosis, does anyone want to ask anything about either of these two things? The reason meiosis happens or the nice outcome we get as a result? It's a good question. He's asking, since it's not a cycle, where are all the parent cells coming from? We'll see this when we get to the second part of the lesson a bit more specifically, but very briefly, in a female, there's a limit. Females only produce so many eggs, and after that, it's over. They run out of cells that are going to go through meiosis. In a male, the cells that start meiosis continually go through mitosis to maintain their population. So even though each cell that's going to go through meiosis can't go through again, there's mitosis happening ahead of it to maintain the population so that production can be sort of endless in that sense. Does anyone want to ask anything else there? Okay. So I have a cell, it's kicking it in interphase where it spends most of its time. Then it gets a signal that it should start going through meiosis, and so we enter prophase 1. Now, a lot of things that happen in prophase happen in both mitosis and meiosis. So I'm trying to highlight some of the things that are different. So even though I didn't say it's happening, the nuclear membrane is going to decompose. Even though I didn't say it again, chromosomes are condensing. So a lot of those things are similar. One thing that's different is that in prophase 1, pairs of homologous chromosomes line up side by side. This process is called synapsis. This didn't happen in mitosis. In mitosis, there were just a bunch of chromosomes wherever. There was no particular order to them, and homologous pairs were not together. So if you look at the picture, I have two pairs. I have a pair right here, the kind of shorter chromosomes, and I have a pair down here, the longer ones. Just like with mitosis, the pictures are representing a cell with very few chromosomes because it's easier to draw. If this were a human, I would have 23 pairs of chromosomes because we have 46 total. Now, when I have a pair of chromosomes lined up side by side, and they've been replicated because the cell was just in interphase, I actually have two chromatids for each chromosome. So in a pair of chromosomes, I have two chromatids on each of them. 2 plus 2 makes 4. So we call it a tetrad. Maybe you remember like Science 10 naming rules, tetra means 4. Maybe you just like the game Tetris and you know that tetra means 4. So if you look, the cell picture, up here I have a black chromosome made up of two chromatids attached to each other, and the red one, two red chromatids. Altogether, I have 4 items, 2 black chromatids, 2 red chromatids. There's a little box around it with the letter T, trying to show you that's a tetrad. Over here, I have just chromosomes pictured, and there's a tetrad on the left of the picture. Now, does anyone want to ask something about a tetrad or what a tetrad is? So in my tetrad, I have a pair of homologous chromosomes. Homologous doesn't mean identical. Homologous chromosomes are the same length, have the same centromere position, but they don't necessarily have the same version of every gene. That's why they're different colors. The picture on the left, they're black and red. The picture on the right, they're green and red, just to show you that there's 2 different versions. When I have a tetrad then, in the middle of the tetrad, 2 non-sister chromatids, homologous but not identical, are next to each other. So if I look at this picture, it would be those 2 in the middle, a red one and a green one. They're the same length, they have the same centromere position, but they're not attached to each other. They're not identical copies of each other. So the ones that are attached to each other, sister chromatids. The ones that are just next to each other, non-sister chromatids. And since they're next to each other, it's possible that crossing over can occur. So if you look at step 2 of the picture here, and I'll just highlight this piece, those non-sister chromatids literally crossed over each other. The red and the green one crossed over. When that happens, they can exchange elements of chromosomes. Depending on where exactly the crossover happens, it could be hundreds, it could be millions of genes. So the result of crossing over is that it can lead to variation. So if we think about what's going on, let's just pretend that the red chromosomes are the ones the person got from their mom, and the green or black ones are the ones they got from their dad. When crossing over happens, some of the genes that originally came from mom and some of the genes that originally came from dad swap places so they could get inherited in a different pattern. If you look at the picture on the left over here, you can see, for example, right there, that crossing over is in the process of happening. Crossing over is probably the most distinctive thing about prophase 1. If I look back over all of the diploma questions that have had prophase 1 as a picture, they all show crossing over. It is like the thing that happens there, but not in other places. Now, does anyone want to ask anything about prophase 1? So, just like in mitosis, after prophase comes metaphase. Now, if you read the description from the beginning, it sounds familiar. Oh, there's a spindle fiber. Yeah, okay. It attaches to each centromere. Okay, I got that. But this time, it's tetrads that we're taking to the equator, not individual chromosomes. In mitosis, the chromosomes lined up one at a time. In meiosis, in metaphase 1, they're going to line up in pairs. So something you might have to do is distinguish between metaphase 1 of meiosis and metaphase of mitosis. And the thing that's different is whether I have one chromosome at a time or pairs of chromosomes lined up in the middle. Now, even then, maybe you might say to yourself, I'm not sure. I'm not sure which one is which. And I just want to highlight this. Hopefully you can see. You can see on the tips of those chromosomes that the red and the black chromatids have exchanged a portion. So you can see that crossing over happened. Sometimes the way you'll know whether you're looking at something in mitosis or meiosis is that you'll see the result of crossing over, which only happens in meiosis. Now, the other thing that happens because they're lining up in pairs is something called independent assortment. Let's just say that the black chromosome is of paternal origin. That means came from the dad. And the red chromosomes are of maternal origin, so came from the mom. In the picture of the cells, there's version A and version B, both of which are possible. What it's saying is, it doesn't matter, really, which one is on which side. In version A, the long black chromosome is on the left, the long red one is on the right. And in version B, it's the other way around. And there's no rule about that. It's completely by chance, totally independent one of the other. And so the second point says the orientation of one pair is independent of the others, meaning just because one pair has the red one on the left doesn't mean the other ones have to have it as well. So which chromosome is on which side is totally chance, random, no rule that indicates which one has to be on which side. Now, because of this independent assortment, there can be even more variation. If you think about this from a human cell perspective, when your cells are getting ready to make a gamete, the chromosomes that you got from your mom, let's say, are not going to all line up on one side, and the chromosomes you got from your dad on the other. They're going to be mixed up. So when you produce gametes, it'll be a random mix of the traits that came from your mom and from your dad. Now that's metaphase 1. Does anyone want to ask anything about what happens in metaphase 1? Yes. Mm-hmm. Usually what I try to do on tests is make them black and white so that you can see, because printing in color is not something we're generally supposed to do. Sometimes the diploma will spend $1 million and print things in color, but I would expect to see black and white so that you can distinguish between the two versions of one chromosome. Does anyone else want to ask anything there? After metaphase, just like in mitosis, we have anaphase. Now you read the first point, spindle fibers shorten. Okay, that's the same thing that happened in mitosis, but here's what's different. Homologous chromosomes are the things that are separating. In mitosis, we were pulling chromatids apart. Here, we're taking one of each of the chromosomes that are in the homologous pair, and we're pulling them apart. You will notice that sister chromatids are still attached. The centromere has not yet divided. And so that's how you can distinguish between anaphase 1 of meiosis and anaphase of mitosis. Now, if you were printing out the pictures, I think I cropped this picture so only half of it showed up because it's really showing the same thing in two different positions in the cell. But the process that could sometimes happen in anaphase is called non-disjunction. In anaphase, chromosomes are supposed to be pulled to opposite sides. If that doesn't happen, we could end up with cells that don't have the correct number of chromosomes. So if you look at the picture up here, it's trying to represent that the cell is in anaphase, but that there's a mistake. That's why it looks like there's a tiny explosion around the chromosomes on the bottom. There's a pair of chromosomes on the top, and they're getting pulled apart. But the pair of chromosomes on the bottom aren't getting separated. They didn't move to opposite sides. In the long run, if that happens, it's possible that the gametes we're going to produce, instead of just being haploid like they're supposed to be, will have the wrong number of chromosomes. Some of them might have more chromosomes than normal because they came from the side that got the extra chromosome. Some of them might have less than normal because they came from the side that didn't get a chromosome during anaphase. Now, we already kind of saw this. There was a question in the textbook the other day about cells that had too many chromosomes. One had all of them, the other had none of them. When you did your karyotypes, the patients all had the wrong number of chromosomes. Now you could add that non-disjunction is what caused it. So when chromosomes, anaphase 1, or chromatids, which is what we'll see in anaphase 2, don't separate correctly, this is what can happen. And I'm going to be more specific about the result. So before I do, does anyone want to ask something about what non-disjunction is? So the picture on the left is really showing us anaphase 1. The picture on the right is representing that in anaphase 2, the second half of meiosis, it's happening. So I'm going to come back to this picture once we go through all of meiosis, because then it'll probably make a bit more sense to look at the second one. So if non-disjunction happens, what's the consequence? Well, gametes with too many or too few chromosomes. You might recall me saying this, but it bears repeating. Usually it's one too many or one missing. Any more than that, and the chances of survival are really basically zero. At this point, we are not really aware of any condition where a person has 2 extra chromosomes or is missing 2 chromosomes and actually is a viable organism, not for humans anyway. So we might have a monosomy. Mono means 1. You are supposed to have 2 of each chromosome. If 1 is missing, meaning that in one of the pairs of chromosomes you only have 1, instead of 2, it's called a monosomy. Turner syndrome is an example of this. Turner syndrome is where a person is female, but they only have one X chromosome instead of 2. So you're supposed to have 2 sex chromosomes, but there is a scenario where a person could have just one X chromosome. Frequently, we'll represent this with an X and a 0 to show that there's not another chromosome there. Now, some of you might remember that in the question in your diploma booklet about fish, this was one of the choices, and a few of you asked me what that meant. Yeah. Mm-hmm. Yep. So generally, having one too few or one too many chromosomes is going to cause developmental delays or abnormalities. In this syndrome, the female with one X chromosome instead of 2 usually is smaller in stature, has mental developmental delays. Not necessarily as extreme as with Down syndrome, but along the same lines. The flip side of this is a person who has too many chromosomes. So a trisomy, where tri means 3, is when there is an extra chromosome. You are supposed to have 2 of each chromosome. If there is an extra one, it means that for one of the pairs, instead of having 2, the person has 3. When we did our karyotype thing on the computer, all of those people had chromosomes missing or chromosomes added, and so you had to identify where there were 3 of them. Down syndrome is a good example. It's where a person has 3 copies of chromosome number 21. So if you looked at a karyotype, you would see 3 on the 21 spot instead of just 2. You do not have to memorize the details of all of these genetic syndromes. They're just examples. Like the diploma question we did earlier this week, we would tell you about the syndrome in the question, and then you would have to identify it in a karyotype, but you don't have to have these all memorized. Does anyone want to ask anything about non-disjunction or anaphase? Then the last step of meiosis 1 is telophase 1. If we take a look at what's happening to the cell, we're going to see reduction division happen here. When we started, our cell had 4 chromosomes. And we are going to move in this phase to 2 cells that each have N chromosomes, where N is 2. Now, if this were a person, a human, 2N would be 46 and N would be 23, but nobody wants to draw 46 chromosomes. So this is the point where we would say officially reduction division has happened. It's the point where the cell goes from being diploid like it was to haploid. This is where the number of chromosomes changes. Now, some cells will not have all of the visible characteristics of this phase, and here's why. In this phase, cytoplasm divides, okay? Then chromosomes unravel and a nuclear membrane forms. Now, if I think ahead, because I know what's going to happen next, the cell is going to go directly into meiosis 2, where it's going to go through prophase, metaphase, anaphase, and telophase again. It almost doesn't make sense for the chromosomes to unravel just so that they can condense again 2 seconds later. Seems kind of silly for a nuclear membrane to form just so that it can break down again. So, when we're looking at pictures of meiosis, sometimes it looks like some of the phases get skipped because they have repetitive parts to them. So once this phase has finished, after it's over, we will end up with 2 daughter cells. So once it's complete, this membrane will fully form between the 2 cells. And I will have 2 daughter cells. I started off with 1 parent cell. Now I have 2 daughter cells. And each of my daughter cells is haploid. There are 1, 2 chromosomes in this one, and 1, 2 chromosomes in that one. You might count and say, no, there's 4. There's not. There are 4 chromatids, sure, but I don't count chromatids as another chromosome in my chromosome number. There are 2 chromosomes in each cell. Now, we're going to go through prophase, metaphase, anaphase, and telophase again, but before that happens, we are not going to replicate chromosomes. So before meiosis started, we were in interphase. Chromosomes got replicated. But we're going to go through these steps again without replicating the chromosomes another time. And that's how we end up still having haploid cells at the end.